Okay, hi folks. I think we're going to get going. I'm not sure how the recording is going to turn out because I'm using both microphones at the same time, so there may be a little bit of feedback, but I'm going to try to manage it. They still haven't fixed sort of the setting in the back of the computer, so we're not able to actually use Zoom at this point, but we will sort of use the OCCS and I'll upload. Do you guys have a preference as to which platform we use, or is My Media or YouTube any better? Either one doesn't make any difference? Do you have a preference? Yeah?
I like YouTube because sometimes my media login has like problems with Chrome or something like that.
Okay, so login issues. Any other preferences or feedback that I can use? No. Oh, wait a second. Yep, did you have feedback?
I would have feedback because usually we open my media incognito, so it gets a little annoying.
Oh, okay. So is it okay to just use both then for the time being? Yeah, okay. Sorry, I don't really have a pin, but I'm ready for you guys this week. I'll be able to hear it drop if I decide to drop it. So there. Yes, I called your bluff. How's that? Okay, so we're going to talk about skin, bacteria on your skin, the correlation. There's only a few papers that are out to actually show causation, but the correlation between skin bacteria as well as skin cancer. And skin cancer kind of runs the gamut in a number of different ways, but we're going to talk about all of those in today's lecture, so hopefully that will make a little bit of sense to all of you. I don't know if you follow like other news articles, but one of the big things that happened over the course of this past week, in the United States, they actually banned red dye number 3, which we will actually talk about later in this course. Canada has decided not to ban it currently, and so we're still using it in different food products. So hopefully that will be something we come back to, talk a little bit about colon cancer. There's been a lot of news about colon cancer this week. Best thing to fight colon cancer according to latest publication, what should you eat or drink? Any idea? We will talk about it later, but that's like after term test 1. Oh, did you hear it? No. Yeah, okay, I heard it too. Milk. Milk is apparently like really great at battling colon cancer, but of course a lot of people don't like drinking milk, and it causes like other gastrointestinal issues, but we'll talk about that later. Today really the emphasis is going to be on things related to skin and skin cancer. Because we have about 2 weeks before you actually are writing your term test, I also wanted to make a really quick note. In the syllabus, the date says that it's February the 5th. However, it's on the same day as class, so it's the 4th. You'll have, I'm going to probably just give you the whole class time. It's really designed to be about a 45 to 50 minute test, but you can use the whole class time if you wish, which is more or less about the hour and 50 minutes. So it's up to you how you decide to do it. You can log in whenever you want, but you can use the entire class time. But it should take you about 45 to about 50 minutes, and to give you at least one of the short answer questions in advance, so you can actually prepare for that and think a little bit about how you're going to write it, and hopefully that will help you a little bit around some of your anxiety. Okay, so we're going to talk a little bit about skin cancer mechanisms, skin and bacteria, as well as some of the glands in the skin, and that's how I'm actually going to be starting today's lecture. And actually, one of the really interesting things is that this is probably the route if you're going to be starting a biotechnology company and you're really interested in skin, because you know that this is like the #1 cancer in Canada and globally, and that you also know that we have the best, easiest access. Like if you're going to go into biotech, or you're going to do a startup, or you're going to be thinking about applications related to some of your other courses, this is where you should really begin. Like it's really around skin, bacteria on your skin, and also like developing through like very, very simple types of genetic manipulations, different types of bacteria that can actually grow and colonize your skin and become potentially those commensal bacteria. And it's actually amazing the types of things that people are doing. So I just wanted to go back and make sure we're all sort of on the same page. Although we're talking about intestinal microbiota here on this particular diagram, in fact, nowadays we call it the GSA, the gut-skin axis. There's also a gut-brain axis, and now there's increasingly a gut-skin-brain axis, because what you have on your skin is literally what you help to ingest, and it does help to form a lot of that original gut microbiome. And of course, that means that lots of things also are going to work to influence your skin bacteria, not just that one-way street. What you actually eat does influence like your skin microbiome. Of course, your lifestyle also will do that. Taking different types of antibiotics will influence your skin microbiome. And of course, hygiene. And one of the things that I've asked you in the last lecture, think a little bit about why after you wash your hands, like your bacteria come right back and you get the same bacteria that you started off with, because they are living in the different pores in your skin and in the different glands on your skin. And I'll try to point that out in the next slide as well. These are all going to be really important in our discussions a little bit later on. And we can also have skin dysbiosis or microbial dysbiosis of your skin. So simplest, most sort of widespread type of skin disorder, we talked about acne. Everyone probably has had acne at some point in time. That's where some of the bacteria are no longer being regulated, and that's where you have dysbiosis. But if we go a little bit further, in fact, dysbiosis can run into like skin cancer, potentially. And that, again, is something I want us to all think a little bit about. So we have normal commensal bacteria, things that normally grow on our skin. We talked about that in the last lecture. We talked about how things can go wrong, and we're going to continue on in that theme. So before we get into that, just to go back and talk about a few things, and again, just for those of you who are taking down notes and for those of you who've joined the course a little bit late, so last week's lecture is going to have pretty heavy coverage. This week's lecture is going to have pretty heavy coverage. And I'm probably just going to use the first part of next week's lecture to finish off a paper. And then the second hour, I'm probably just going to answer questions and then give you a couple of questions in advance for you to think about. Does that sound like an okay plan? Just so that there's not too much material last minute for all of you to be thinking about. And I talked about this really quickly. In fact, we're not going to be covering mammary glands. It is a type of skin-associated gland, but we won't be talking about them. We'll be talking about the first four, the apocrine, merocrine, ceruminous, and then the sebaceous. So the sudoriferous or sweat glands, these are widely distributed along the body. We have two different types of these sweat glands. The composition of what they are secreting tend to be a little bit different. For the most part, though, it's mainly water. There are some salts that are associated with it. We also have some metabolic waste that are eliminated through the sweat. One of the things that's really important about your sweat is that it actually helps to create what we call an acid mantle on your skin. And this acid mantle on your skin is important because that slightly acidic pH of about 4, for some individuals all the way down to an acidic pH of 4, all the way up to about a pH of 6 actually does inhibit bacterial overgrowth. So we don't want to have too many bacteria growing on your skin. And in fact, your skin has designed a number of different types of secretions, this being one of them, to actually help maintain that commensal level of bacteria. Okay, so these sudoriferous or sweat glands, really important. And the two subdivisions, one, the merocrine sudoriferous gland. The older name for this is the eccrine sweat gland. This is the most numerous type of gland on the skin. So we find these on the palms, the soles of the feet, on your back and chest and other areas. These are normally associated with tubular outgrowths, and these are simple epithelial tubular glands. So this is that type that produces the watery perspiration. If it's a hot day and you're sweating a lot, your merocrine glands are working overtime to produce that watery perspiration, very helpful in maintaining that body temperature. And those simple epithelial cells also are myoepithelial cells, so they have myosin associated with them, so they can actually contract. And the contraction actually allows the water and the sweat to be pushed upwards, and they respond to things, different signals from the sympathetic nervous system. Okay, so hopefully, really quick overview. I'm not sure if I covered that in the last lecture or not. It's sort of a vague recollection that I kind of did, but I'm not sure. Yes?
You just introduced the 5 different types, but we didn't go into.
Okay, so this again would be one that you should probably know for the upcoming term test. And for the term test, the next few slides, you're probably going to get somewhere around 2 to 3 multiple choice test questions. Hopefully it's a straightforward multiple choice style test for all of you. I will give you some sample questions in advance, and we will have sort of a review next week in the second hour. Just sort of as an overview of what this looks like. So this would be sort of one of the different types of glands that we have. Those would be the myoepithelial cells. These are capable of contracting, and they will be pushing up the sweat and the watery sweat up through this duct that's associated with it. And again, it's one of those really important types of glands that we have on the skin. And this is that miracrine type of sweat gland that I just mentioned, that subclassification of the sudoriferous type. So again, before I go on, because you're also a very quiet class, any questions? No, none. Okay. I will take your silence as you're all okay for the time being. So on that previous diagram to the left, we also saw these other types of sweat glands, and these are the apocrine sweat glands that you are seeing here. These are a little bit different. They're much more specialized. They're found in areas like your armpit or the axilla, in areas around the groin, and areas that have a little bit more hair. Most of these ducts empty into glands, or are glands that empty into like hair follicles. And the sweat that's produced here is a little bit thicker than the miracrine type, which is more watery. It's not as clear. It tends to be a little bit more colored. It looks milkier if you're able to collect enough of this. I would advise you not to do that. It's a little bit milkier because it also contains fatty acids and other secreted types of proteins. These glands tend to develop a little bit later. So we all start off with all of those miracrine types of sudoriferous glands. The apocrine glands usually start appearing during puberty, and then they persist throughout adulthood. And the type of sweat that is produced by these apocrine sudoriferous glands are involved in what's called bromhidrosis. And this is where your sweat actually doesn't smell so nice. So if you've ever smelled sweat and someone has like very odory sweat, it's because of this bromhidrosis. It's where the bacteria on your skin are taking some of those different types of fatty acids in this type of sweat, breaking it down, and the byproducts, the things that smell bad, the byproducts of that breakdown by your skin bacteria are what produce that odor. So all of those different types of things, again, showing you that there is a normal physiological interaction between bacteria and your normal physiology. Yes?
Is this the way, is this like, is the skin essentially like feeding the microbiota, or not the microbiota, but
the bacteria?
On your skin? Or is this?
Yeah, so in a lot of ways we know, for example, from last week's lecture, when we talked about the paper by Lee and Fitzgibbon, we know that we will start having an overproduction of certain types of bacteria, and yes, they actually digest a lot of the different byproducts of like dead skin cells, the byproducts of sweat and other things. So yes, they live there, and they can continue to live there because they're digesting different things. Okay, so this one I actually did some homework on. So these are ceruminous glands. These are only found in the external ear canal. And this is where these gland secretions, the ceruminous gland secretions, actually combine with something that your skin normally produces. And if you've ever gone and bought like a skincare product, you'll often see control sebaceous secretions or control sebum. Don't know if you've ever read that on some of the different skincare products. But the oil that your skin produces is actually called sebum. We're going to see sebum again a little bit later. And the ceruminous gland secretes additional factors, and it combines with the oily secretions from other epithelial cells and the dense skin cells to produce earwax. So when you're thinking a little bit about ceruminous gland secretions, this earwax is actually something that gets produced. That earwax, again, is waterproof, so it helps to keep the eardrum flexible. It also prevents other types of organisms from entering into your external ear canal and then potentially damaging your ear or just being an irritant. I'm not sure if you've ever had something trapped like in your ear before, like an insect. It's really hard and it's very, very annoying if it gets in there. But you don't often get insects kind of flying into your ear because apparently, and I can actually attest to this, I'll tell you that story in a second, earwax is bitter and the ceruminous gland secretions are bitter. And they help to repel anything that could theoretically crawl into your ear because it doesn't really taste good, and those organisms can actually sample that. So this also will inhibit bacterial growth. So the ceruminous gland secretions that include like sebum, dead skin cells, etc. And that sebum and that wax help to keep the guard hairs around the outside of the external ear canal a little bit sticky to, again, to produce a barrier for things like dust and other things to enter into your external ear canal. And I had asked my wife if I could get a sample of her earwax, and she said no. And I was quite surprised, and she asked, like, why do you need to dig into my ear? And I said, well, it's for a class. I need to do some research. And she said, don't you have 2 ears? Can't you like just dig through your own ear? And I said, hadn't thought of that. She goes, okay, do your own experiments on yourself. And so I can tell you that apparently my earwax is not very pleasant tasting, a little bit bitter. Sorry, don't visualize it. Just take it for granted that yes, it actually works. It's not like I sat there and ate the whole thing, you know? So, okay, anyways, I did my research. It is bitter, and we'll just leave it at that. Okay, we'll move on. We'll talk about sebum now, something a little bit more interesting, I guess. So these sebaceous glands are found over the entire body, except on the palms and soles of your feet. So this is where that oily substance, that sebum that we saw on the previous slide, actually comes in to play. It is an oily substance. It's classified, these types of glands are classified as simple alveolar types of glands. And this is a form of what we call holocrine secretion. And I'm not sure if you still learn these terms in like PSL 300 or 301 when you do the endocrine system. Is this ringing a bell at all, like holocrine secretion? No? Okay, it's not really important. This is where the entire cell basically self-destructs. And as it does so, it actually helps to form part of that secretion. So the holocrine part is literally where the cell destruction leads to the secretion itself. And this is more of an exocytic type of secretion as well. Again, also most of these are associated with the hair follicle, so it's similar to the apocrine type of secretion that I mentioned before. And we need that sebum. It does a lot of different things. One, bacteria like to eat sebum. That's not on here, but we learned about that from last week's lecture. So bacteria do ingest sebum, and they will divide more quickly if there's more sebum around. But that sebum is there on your skin normally to collect dirt. It's also there to kind of lubricate and soften your hair up to a certain point. Like you don't want to just keep getting more and more sebum on your hair. You do want to wash your hair at some point as well. But for the most part, it is there to kind of make sure that it is lubricating and keeping your hair soft and lubricated. Okay, so just again, as another reminder for all of you, and I'm just trying to think to make sure I'm not saying anything inaccurate. For your term test, not next Tuesday, but the following Tuesday, you don't really have to worry that you're going to have to label a diagram or identify a part in a diagram, so you don't need to worry about diagrams too much. But the bullet points, wherever you see them on a slide, those are ones that I would really make sure that you have understood, connected. For example, on here, I don't have sebum from the previous lecture on here, but you might want to make that link that sebum was something we talked about the week before. Okay, so just to make sure that we're all sort of on the same page. Any questions on that or comments? Things that I've gone through too quickly? Still trying to get that thought out of your head on ceruminous glands and things? I'm hoping not. So we're going to switch gears a little bit. So we've been talking about skin. We talked about glands on the skin. That's going to be, again, important a little bit later, but it was sort of also a link to what we talked about in the last lecture as well. So the current sort of guidance from different health organizations is that you should never ever get like any type of sunburn ever in your entire life. For many of you, and including myself, you probably have experienced a sunburn at some point, but there's increasing evidence to suggest that at some point it's going to not be so good for you. Also, as you get older, as we'll find out later on today, your immune system function within your skin actually dramatically goes down after the age of 50 and really drops off by the time you're about 65 to 70. It's like well described in the literature now, so your immune system is really bad. And that also means that for your skin, that means that sun exposure later in your life is also not great. So for your parents, for example, getting too much sun exposure without protection is actually not a great idea. They're more likely to develop skin cancers later in their life, not from an original incident that happened when they were like in their teenage years or in their early adulthood. It's really later in life. But the overall guidance is still, you should never get a sunburn. So these would be like different examples of like a first degree sunburn, like you just turn really, really red. And I think some of you probably have had that. I know I've had that in the past, like I just forgot to apply sunscreen. I went out, I forgot the back of my neck, and the back of my neck like turned completely red. Guys have probably experienced that at least once, right? And that's where the upper layers, particularly in the epidermal layers down to the dermis, almost down to the dermis, are completely burned. If we talk about a second degree burn, this is going through the entire layer of the epidermis down into the dermal layer, which we've talked about, and not quite to the subcutaneous layer, but it's getting there. And then in terms of a third degree burn, this is the full thickness of your entire skin. So it's epidermal, all the way down to the dermal, all the way down to the subcutaneous layer. This is serious. So like this typically doesn't happen unless you have a genetic disorder where you just don't have pigmentation and you've gone out into the sun. You wouldn't get a third degree burn. Even a second degree burn, it would start hurting a lot. So it's very unlikely that you get a second degree burn. But a first degree burn, as you see here for this individual, pretty common, even with sunscreen on unprotected parts. So as you're getting exposed to sun, and as you are getting that sunburn on your skin, you're being exposed to UV radiation, and this is actually very serious. So we're 100% sure that UV radiation exposure actually can cause skin cancer. In addition to that, every time you're being exposed to UV radiation, when you're in the sun, for too long a period of time, it actually breaks down folic acid, which I'll talk about in a moment. And folic acid is actually something that's essential for normal cell division. It's particularly important if you're pregnant and you have like cell division at a very rapid level, especially during early pregnancy, for example. You need a lot of folic acid. So if you're getting too much UV radiation when you're pregnant, it's also really not great. Not great for you and also not great for fetal development. But again, I'll talk about that later. Last week's lecture, we did talk about the beneficial aspects. Like we do need some sun and we do need some exposure to UV rays. Although again, really controversial, like do you really need sun exposure at all? Because nowadays with different types of supplements, vitamin D is something that you may not need to make through your skin, and it may not be something as important as we used to think. And there's sort of a rethinking that perhaps in terms of the guidance that different individuals are getting, it's really not sun exposure that you want. It's like different types of wavelengths of light that you should be exposed to. So I think I mentioned in passing, although I could be wrong, one of the things that's really important now in terms of biology is like looking at light therapy, both for mental health as well as for like recovery from different types of ischemia, recovery from different post-workout. Red light therapy seems to be really important. Yellow light therapy seems to be really important. Maybe we don't need UV in the sunlight as much as we used to think. We don't really synthesize too much vitamin D directly through our skin anyways. So maybe we don't need to have that sun exposure for for vitamin D production. And again, in terms of the length of sun exposure and UV exposure, so individuals who have a background where they're more in the tropical zones, they have more melanin in their skin, and we talked about that way back in lecture number 1. And this melanization of skin and darker skin tones tends to be protective against different forms of excessive UV rays. It doesn't mean that they can't develop melanoma. In fact, they can, but it's less likely that they're going to have some issues with that. Individuals who are more fair-skinned, who have originally come from cultures that are in the far northern or southern latitudes, sunlight tends to not be as excessive and tends to be weaker, so they tend to have lighter skin to allow for more UV penetration. But of course now with globalization, it's really not as big an issue. So one of the things that gets broken down when you have too much UV exposure, and it's UV, it's not sunlight, it's UV exposure, is folic acid. And we need that during like early development. Usually because your cells are dividing at such an incredible rate, somewhere on the order of around 40,000 cells per minute to 250,000 cells per minute, depending on the area that you're looking at during like early embryogenesis. It's a lot of DNA replication that's going on during early fetal development. So within the first trimester, there is tremendous growth, like 250,000 cells per minute, like every single minute. It's dividing again. It's an incredible amount. So during early pregnancy, we need excess amounts of folic acid. So usually this is taken before a planned pregnancy. And this is, again, vitamin B9. It's a water-soluble vitamin, often now taken as a supplement. And this is essential for DNA replication. And this is also really essential, folic acid or vitamin B9, also essential for the synthesis of proteins. And if you think about how rapidly a cell is dividing, and you're creating like 250,000 new cells every single minute during early embryogenesis, that means that there are a lot of new proteins, there's a lot of DNA, there's a lot of folic acid that's going to be required. And unfortunately, one of the areas that is very vulnerable to this, for those of you who've taken embryology or have taken like HMB322 with me last week or the week before, you'll know that folic acid is actually really essential during neural tube closure. So when your spinal cord is actually finishing its overall structural formation, the neural tube is one of the areas that has to go from being sort of a flat surface, then it has to invaginate, and then it will completely break off and bud and form a tube-like structure. And if you don't have folic acid during that period of division, that actually doesn't happen. And you're left with some continuity where you have a protrusion out of your back of an incomplete closure of the neural tube, and this often results in spina bifida. And unfortunately, that's not so rare. It's not so rare. You can actually see spina bifida quite frequently. It is something that can be fixed through surgery. Anencephaly, though, is where the front part of the neural tube, not the back posterior part, the anterior part of the neural tube fails to close. When the anterior part fails to close, that means that your brain basically doesn't form well. And unfortunately, that leads to what's called anencephaly. And that's usually lethal, like the embryo doesn't survive. And so one of the thoughts that different individuals have had is that is melanin really designed to kind of shield the nucleus, the DNA in the nucleus from any kind of degradation? And is melanin also designed to shade some of the folic acid that would have to enter into the nucleus to be able to help with that division? So again, it's really fascinating, but it tends to be like borne out in terms of both epidemiology and in terms of the biology that folic acid does break down fairly easily with direct exposure to UV light. And unfortunately, if you don't have enough of it, and the mother is the source of that vitamin B9 or folic acid, it does lead to neural tube defects. Most commonly spina bifida, but also anencephaly. So how many of you have heard of like your actual age versus your skin age? Like, you know, you all have your birth date, you can calculate your actual age, but then depending on how much sun exposure you have, your skin might appear older than it should. And in fact, we can actually tell how old your skin is by looking at some of the different types of photoproducts that are in different parts of your skin. So when you get exposed to UV radiation, if you've been sitting out there in the sun too long, it actually damages your DNA. Sometimes it can be repaired, sometimes it can't. So DNA damage to some of the different keratinocytes. And you have to think about this, and I'm going to ask you to think about this. And I'm going to ask you to think about this before you get to the first term test. Do keratinocytes normally have nuclei associated with them? Okay. I like the fact that we're now moving to hand signals. We're not speaking, but we're moving to hand signals. Love it. They don't, right? But there are some keratinocytes in certain levels of the skin or certain layers of the skin that do. They do have a nucleus. You might want to think a little bit about that, because when we're talking about DNA damage to keratinocytes, which keratinocytes actually have those nuclei? And again, getting you to do a little bit of that background thinking before the term test, because you have like the full hour and 50 minutes, but you don't want to be kind of leafing back through your notes back and forth. And those melanocytes as well. And depending on where the DNA damage occurs, it will either result in like a basal cell carcinoma, a squamous cell carcinoma, or if it's a melanocyte that is aberrantly dividing, it can lead to a melanoma, which we know is the most dangerous type of skin cancer. So when you're exposed to UV radiation by sitting out in the sun for too long without that sunblock, it can actually produce mutagenic photoproducts in your DNA. And these photoproducts are found in adjacent, side-by-side pyrimidine base pairs in the DNA. And when that happens, it actually produces something called a cyclobutane pyrimidine dimer. So it creates these little dimers, which you're seeing here. And again, you're not going to have to worry about drawing out a structure or identifying a structure like on a diagram, but I would like you to make sure that you know these bullet points. And this is a signature mutation that is found in your skin if you have been exposed to skin and you've been exposed to a lot of sunlight on your skin. So if you have too many of these in dividing populations of your keratinocytes, you can actually detect them. And when you detect them, you know you've actually damaged your DNA. And depending on the types of pyrimidine dimers that are actually formed between these adjacent pyrimidine base pairs, you can have different types of mutations, which are on sort of the next slide. So these are the two pyrimidine base pairs, either a T or a C. And if you have two adjacent TT base pairs and you form this type of cyclobutane dimer, this one tends not to be as dangerous. It's not quite as mutagenic. However, if you have them with the CC base pairs and they're right beside each other, these are often more tightly associated with mutations in your DNA, whether it's in the melanocytes or within the keratinocytes that are nucleated, that lead to different forms of cancer. So we'll be talking a little bit about DNA polymerase, the base checking function, what happens with aging, and the ability for DNA polymerase and the correcting ability for different DNA polymerases also goes down with aging, as does cellular homeostasis. A lot of things just go downhill in aging, and so you're more likely to develop different types of diseases and cancers with aging. And this is where you start seeing the mutations coming in a little bit more pronounced, and this will be sort of the last lecture in this series before we move on to talk about other things. If you were to get that cyclobutane CC pyrimidine dimer, and it happened to happen in an area of your DNA in either your keratinocytes or your melanocytes, and it was in an area of your DNA that involved like the p53 tumor suppressor gene, this could be one of the most important triggers. In fact, for many forms of very aggressive skin cancers, this seems to be the case. You've developed this mutation in the p53 in certain types of cells. Again, this p53 tumor suppressor gene is something that we'll come back and talk about, but as the name suggests, the ability for you to kind of maintain tumor suppression through the normal function of this gene is going to be lost if we have these types of cyclobutane dimers forming. In addition to that, again, one of the other things that's really important about this, the multi-hit hypothesis around cancer still is in play here. So it's not just a mutation that's allowed to continue and not get removed. It also means that your immune system also has to not come into play. And UV radiation also affects these immune cells, and it suppresses a lot of the immune response, and it induces immune suppressor T cells when you're exposed to too much UV radiation. And so as a result of that, you're also going to have, as a byproduct of activation of these T cells, you're also going to be releasing a number of other pro-inflammatory cytokines, things like interleukin-1 alpha. Interleukin 10, again, seems to be sort of up and down a little bit, so you're going to get a little bit of a reduction in interleukin 10, which is normally an anti-inflammatory cytokine, and you're going to increase the levels of TNF alpha. And so this creates sort of a pro-inflammatory state within your skin, which might lead to the development of skin cancer. So again, this is sort of older data. The newer data from 2 years ago suggests that even one single sunburn, a severe sunburn, leads to an increased potential in developing skin cancer. But as sort of a take-home message, the more sunburns you have, the increased risk versus an age-matched population that you're going to have for developing like malignant melanoma. Sun exposure increases that by about 4-fold. Squamous cell carcinoma, that goes up by about 2.7-fold, and basal cell carcinoma goes up by about 2.6-fold. So regardless, having too much UV exposure leading to damage on the nucleated epithelial cells, the keratinocytes in your skin will eventually lead to these different types of skin cancer. Any questions on any of that before I go on? Yeah, please.
By blistering, do you just mean like when your skin like peels, or is it like severe blistering, like a second-degree burn?
Have you ever, so I'm not sure if you've ever had a sunburn. So if you've ever had a sunburn, one of the first things that happens is that it turns red, then you start to see a little bit of blistering. It kind of raises itself up, and you know that that part's going to start peeling off. And that's what it's talking about here. We're not at the level of like second degree where we literally will have like blistering as one of the first things that you see in your skin. So it's part of that continuum. And for whatever reasons, I'm just thinking about my wife who loves when I get a sunburn because she can't resist peeling that peeling skin. Sorry, we have a weird family. That's all I can say. Don't tell her I said that. Okay, so if you don't have any other questions, I'll just kind of talk a little bit about the epidermis. We need to talk about a few very essential things. Again, there's a lot of coverage from this lecture, and I think I've already pointed out that you have about 15 multiple choice test questions, 2 short answer questions. It's likely that one of your short answer questions is going to come from this lecture, so you might want to make sure that you're taking down some good notes, sharing some notes with your friends a little bit later on. And I'll give you some examples of what I mean by a short answer question. They're not designed to be like 5 or 6 sentences. They're 2 or 3 sentences, very short. Some of them might just be 1 sentence, like what might happen if this cell was exposed to this. Like they're designed just to test whether or not you've understood and can apply different concepts in this course. And I'm assuming a lot of you will do really well. So hopefully you won't let me down that way. So we've been talking about the epidermis. We have 4 or 5 layers. These are all stratified in terms of the epithelial cells. The major cell type that we've been talking about are these keratinocytes, most of them on the apical surface, on the one that you see the most often that flakes off. They're enucleated, so they no longer contain a nucleus. However, the basal keratinocytes, the ones that are especially attached to the basement membrane, which is the membrane that separates the epidermal layer from the deeper dermal layer, they do have a nucleus, and these basal keratinocytes remain undifferentiated, and they can continue to proliferate And again, they are like these stem cells, and they can continue to divide, and they're the ones that will continue to divide. As they move away from the basement membrane layer, these cells start to differentiate, they leave the cell cycle, and then they become enucleated, and eventually they will be lost from your skin, and we've talked about that before. And again, in terms of UV exposure, just as a review, so we know that UV light is important in that sun exposure. It can also activate keratinocytes, which I'll talk about in a moment. We've talked about these photo products. It's a good idea to make sure that you know the difference between the CC and the TT cyclobutanes, why that's important, why we look for them in different keratinocytes, what keratinocytes in your skin. Again, I'll leave that to you to kind of do a little bit of homework. And then one of the things that we'll see is this P53 pathway actually does come into play in your keratinocytes. So we don't want damage to this to begin with. And I'm going to be taking you through over the next few slides on some of the different ways in which your skin normally responds in an appropriate physiological way to sun exposure. So while it's not good for you, your skin is trying its best to adapt. Like the reason why you're getting tanning is not just to look nice, is it literally is trying to save your DNA by trying to shield it with different types of molecules and that melanin that we talked about to absorb some of that UV radiation. One of those molecules that is produced by your keratinocytes is melanocyte stimulating hormone, or MSH. It's going to be released by the different keratinocytes that are being stimulated by UV radiation. This will then work on nearby melanocytes and the M1 receptor. And when we look at that on the next diagram, you're going to have to tell me what type of receptor that is, or what class of receptor. And ultimately, in the melanocyte, after activation of the M1 receptor, this is going to lead to the increase in this factor called MITF. And this will lead to the production of things like melanin, which then gets spread to other nearby keratinocytes. Okay, so that in a nutshell is where we're going, and I'm going to now take you through that step by step. Before I get going to the next slides, do you have any questions or things I can clarify for you? Yes?
Keratinocytes self-renew or no?
Yes, they're capable of self-renewal. Like they're basically the source of all your skin for your entire life. I'm not sure if there's another, yes?
So basically, tanning is just increase in melanocytes because
It's not an increase in melanocytes. That would not be a good thing. No.
then like I'm kind of confused, like
Yep, I'll show you that in the next few slides. Yep.
Sorry, you mentioned that the basal cell keratinocytes could become cancerous. Can they still differentiate after
Okay.
once cancerous, or?
Once they're cancerous, no. No. Once they've kind of decided that they're cancerous, no. Any other questions? Okay, I'm hoping the next slide is the one I have in my head. It is. There are a couple of things that are on here. You might want to jot them down. It's up to you. Like I'm not, I'll give you like both the long form and the short form wherever I can on these different questions. But it's up to you if you need to kind of write out some of these different things, and I'll kind of take you through part of it right now. So you're going to have to imagine, and you're going to have to think about this as a schematic diagram. So it's it's not completely realistic, but it's pretty close to it. So this would sort of be the level of the stratum basale. Do you remember the stratum basale? Do you remember what else it's called? Going back like 1 week to potentially 2 weeks? Yep.
Oh, stratum germinativum?
Yep, stratum germinativum, and again, germ cells, dividing cells, basal cells, however you want to think about it. We're looking at the very base level, right between the last layer of the epidermis, and underneath we would see like the dermal layer. We don't see the basement membrane, but it would be here. Here, in blue, we have one cell type. This is the melanocyte. Okay, are we clear on that? This is that one melanocyte that we have here. This is the nucleus of the melanocyte. Good so far? Schematic diagram. Beside it, we have these three other cells. These would all represent nucleated keratinocytes that all contain DNA. So this would be one keratinocyte, this is another keratinocyte, all sort of at the stratum basale, and this is the third keratinocyte. Do you follow along with that? So we have two different cell types, melanocyte, and we have these keratinocytes above them. And again, we've been talking a lot about UV exposure. So you're sitting there in the sun, the sunlight contains UV rays, the UV rays are now penetrating through your skin, and they are now hitting the nucleus of these keratinocytes. What is the most abundant cell type that we have in our skin? It's not a trick question. Where's my pen? I lost. Yeah, keratinocytes. Most abundant protein, keratin, right? Keratinocytes. Okay, so we have a lot of keratinocytes. We have a lot of keratinocytes being exposed to UV. The UV radiation comes in and it will actually cause the DNA to be a little bit damaged. And one of the things that results is the transcription of this factor called ATR. This will activate the P53 gene pathway, and this in turn will eventually cause the production and the release of MSH, melanocyte stimulating hormone, from this keratinocyte. Okay, so this is that one keratinocyte. Are we okay with that so far? So we're looking at this one keratinocyte, its nucleus has been exposed to UV. As a result of that, you've activated ATR, which is the short form for this long named gene. You turn on the P53 pathway, and will also activate the POMC pathway, and eventually you'll form melanocyte stimulating hormone, which then gets released from this one keratinocyte. In addition to that, before I go on and then we'll take a break, in addition to that, this UV radiation will not only from this one keratinocyte, so we're still looking at the same keratinocyte in this little bubble down here at the bottom, still a keratinocyte. We're also going to be releasing or causing the release, because we have turned on the ATR pathway, not only melanocyte stimulating hormone, which we see here, we'll also be releasing other factors like basic fibroblast growth factor, BFGF, and we'll also be releasing stem cell factor, or SCF, which you see here. This is going to be important in the next diagram when we come back from break. But this single exposure to UV radiation on the keratinocyte actually activates genetic programs, which will then cause this keratinocyte to release all these different types of proteins like MSH, like BFGF, and stem cell factor, which is what you see here. And these will then be targeted to other nearby cells. And I'm going to stop there, and I will pick things up after the break. And if you have any questions, I'm happy to answer them. Okay. Just a really quick note as well. Some of your colleagues have actually created sort of a group chat, and you can indicate who you guys are. So if you want, you can join their group chat to study and to kind of do other test questions and things together. I'm also going to be posting the group chat on Quarkus later. So if you don't get it from them today, you'll see that later on Quarkus as well. So hopefully you guys can kind of build community in your class wherever possible. So we've been talking sort of about the left side of this particular diagram. We've been looking at the production of melanocyte stimulating hormone, MSH, from this cell that's been exposed to UV radiation, and how this MSH and this stem cell factor, or SCF, that has been released from a genetic program from the damage that the cell has received after UV radiation has hit it, is now going to be taken up and signal to the nearby melanocyte. Okay, so far we've only been looking at the keratinocytes. So far there's nothing related to tanning, nothing related to melanin formation. But the next part of this kind of relies on the first part that we talked about, the MSH that's being released, the stem cell factor that's being released, among many, many other things. This cell, by the way, is also likely just going to die through apoptosis. And again, if it is able to repair itself, it will activate XP genes, but it depends on the level of DNA damage, which DNA got damaged, etc. But this cell can also undergo apoptosis. So let's follow this cell now. This is the melanocyte, and the melanocyte is what we're going to look at here. And the melanocyte is something that, for example, has on its cell surface this receptor here, which I told you on the previous slide is the MC1R. If you were going to classify this type of receptor, what would you classify it as? Yep.
Is it transmembrane protein channel?
It is a transmembrane protein channel, more general, like bigger. Yep.
Well, I was going to say it's a G-coupled protein receptor.
Yep, so this is a G protein coupled receptor, heptahelical receptor, 7-transmembrane, whatever you want to call it. This would be like a GPCR, and the MC1R is a GPCR that when melanocyte stimulating hormone binds to it, or MSH, that's being produced by the nearby keratinocyte, will then cause the activation of adenylate cyclase, and then it will increase intracellular levels of cyclic AMP, which in turn will activate protein kinase A, or pKa. And pKa, its main function here in this melanocyte is to cause the phosphorylation and therefore the activation of this transcriptional factor called Krebs. So the formation of phospho-Krebs is something that you'll see after exposure of the melanocyte to MSH. So before I go on to the rest of this pathway, are we okay with that so far? And one of the ways I kind of like asking questions is more like application. If someone has created a substance that will bind to the MC1R receptor, assuming that it works the same as MSH, which of the following might you see an increase in the level of? And you would be able to go through some of these different pathways. So ultimately, the importance of this is that the phospho-Krebs will then activate and will sit on different regions of the gene and cause the transcription of MITF in this melanocyte. Okay, so phospho-Krebs is a transcriptional activator. It turns on the expression of different genes. One of the gene products is going to be MITF. Are we okay with that so far? In addition to that, because this cell has received cell damage, and this is the keratinocyte, it also releases stem cell factor, which is what you are seeing here. Stem cell factor is also critical to this pathway because stem cell factor interacts with the melanocyte in the same way, and it activates the c-KIT receptor. It is not a G protein coupled receptor. It's very different. It sort of has to form a dimer, so it dimerizes, and after dimerization of the c-KIT receptor, it autophosphorylates, which is what you see here. And this autophosphorylation, in turn, will cause MAP kinase to then phosphorylate the MITF protein that has been produced through the other MSH pathway. So that MITF is now going to be phosphorylated. And you might think, well, this is really complicated, but it's actually really necessary because this phosphorylated MITF, or MITF, is now itself going to act as a transcriptional regulator, and it will turn on the transcription of a number of related genes as well, all the ones that you are seeing here. And the most important part about all of the genes that it is turning on here, and I'm not going to get into all of the different details, but these are all parts of the amino acid pathway, things like tyrosine, tryptophan, and others that will eventually, as you see here on the right, which again, you don't need to memorize anything in this pathway, ultimately, all of these genetic programs act to increase the formation of melanin from this melanocyte. Does that make sense to everyone? So the pathway here in panel D is not one that you need to memorize. And again, I'm not going to go through all of the different genetic programs here on the right or the bottom right on panel C, but these are different things that will eventually lead to the formation of increased pigment or increased melanin, which then gets packaged into these different melanosomes And from lecture number 1, we know that these melanocytes have these long projections that will then contact a number of different nearby keratinocytes. They will be releasing these melanosomes, which are then taken up by the nearby keratinocytes. And as they're taken up by the nearby keratinocytes, this melanin can now act sort of as a shield and protection from UV radiation in these other nearby keratinocytes. Hopefully not too convoluted. Does that all make sense? Is there anything that you need some clarification on, or things that I may have gone through too quickly? Yep, please.
So with the stem cell growth factor, the dimerization of skip, so it leads to the MAPK kinase, like MAPK pathway to be activated,
Yep.
which then
Phosphorylates.
phosphorylates it, and then it leads to a transcription.
Yep, so this MIT-F, phosphorylated MIT-F, will then act as a transcriptional activator and turn on other genetic programs that lead to the formation of more melanin and package more melanin into these melanosomes. And I'm not going to go through these different pathways. And you don't need to memorize the synthetic pathway here on the right, but it's
So
basic.
They both basically work in tandem.
They need to work together, that's right. Any other questions? Yep.
I have one more.
Yeah.
Can it work as they're working tandem? But can it work like just the MSH or just the skin or the stem cell growth factor, or they both have to be there to activate it?
So this is the most effective way to actually increase the levels of melanin in your skin after UV exposure. It's why your skin gets darker when you go out. You're producing more melanin. Can it do it without sort of the activation of the c-KIT pathway? It takes more time and it would be less efficient. In theory, this MIT-F can be phosphorylated by a number of other types of kinases, but it also has a fairly short half-life, and so it would have to encounter another kinase that's capable of phosphorylating it, and this pathway is one that's already available to it, and it's already at work. Yep. Okay, any other questions? No. It's kind of really terrible to be talking about tanning warm weather on a day like today, I know, but there we have it. Yes?
So those who naturally born with higher melanin
Yeah.
amount,
Yep.
is this the mechanism without the UV that goes into producing more melanin, or are there other?
Nope. So you have a genetic program that would already be activating in your melanocytes an increased production of different types of melanin for individuals that have darker complected skin. But you yourself, when you're getting exposed to more UV, will increase the levels of your own melanin. So you will still get tan.
Okay, and then so the reason why it's temporary, I'm assuming, is because these cells eventually go to the top layer and then die.
Yep. So eventually they will be lost and we won't have that pathway available. These cells also die. Yep. It would be great if you could have a tan that lasts forever, but it doesn't happen. Any other questions? No. Bless you. Okay, so again, just a few more things. So the c-KIT receptors that we just talked about, it's going to be activated by the stem cell factor. This leads to the activation of MIT-F, and those are all pathways that I've just been talking to you a little bit about. So hopefully that's, so both the start before the figure and then the figure itself, and now this sort of wrap up bullet point, hopefully they all make sense and they kind of explain the pathway that we've just gone to. So one of the things that is increasingly found nowadays, I guess, in dermatology is actinic keratosis, and more people are kind of looking at this as a form of precancer. So this is one of those markers in your skin that we've been kind of talking about off and on. Like, your skin is a great diagnostic tool. And it's really hard because sometimes you just have things that kind of look like eczema, or you have things that look like a rash on your skin, and it's really hard to kind of know. Or sometimes you have things that feel like your skin is a little bit more scaly. And this actinic keratosis is now thought, like this type of skin disorder is thought to be a form of pre-cancer. And again, just to put things back in perspective for all of you, now that we've kind of talked a little bit about the issue with UV light and the fact that UV light, we have a normal physiological response to protect our DNA and the keratinocytes. Skin cancer is still the most prevalent cancer globally and in North America. You are all going to see an increased rate of skin cancer over the next 20 to 25 years. And unfortunately, we don't have a lot of great sort of outcomes on the horizon for all of you. One of the things that we do know, and we've been talking about in today's lecture, in previous lectures, one of the things that is a predisposition for skin cancer is UV radiation exposure, sunburns, etc. This is a very well-established risk factor that we talked about before break. And there is emerging evidence to suggest that the human microbiota, the bacteria, and other organisms that live on your skin may actually influence skin cancer development, which again is very, very interesting. Probably in the last 10 years, there's been the most research related to it. And again, we already know things like acne, the most common sort of dermatological issue, that is directly impacted by changes in the different types of bacteria that live on your skin. Things like atopic dermatitis and changes to your skin texture, all of these things are affected by different types of bacteria that live on your skin. So I'm just going to introduce something that some of you may see in the literature if you're doing skin cancer as one of the types of cancer that you're going to be following up on. The non-melanoma skin cancer just encompasses both of the two other types, the squamous cell as well as the basal cell carcinoma. One of the bacteria that seems to have a high prevalence, we see it more tightly associated with squamous cell carcinoma, is finding more Staphylococcus aureus. So Staph aureus seems to have a very, very high sort of likelihood of being found within squamous cell carcinomas. And you might be asking, like, is it the cause, the effect? Is it just like a natural byproduct? There's a lot of evidence to suggest it might be directly involved. And there's also some suggestion that it's also the byproduct of having a cancer on your skin. So again, we're not 100% sure yet, but actually it's just really amazing to think about the research that's been going on in the last 10 years related to the microbiome and skin cancer. So one of the reasons why this is important, we have Staph aureus normally growing on our skin, and this colonization is very tightly linked to that precancer that I talked about to you before, actinic keratosis. The other thing that is also associated with Staph aureus on your skin and growing on your skin, not only does it probably form actinic keratosis and is very tightly regulated to that, but it's also shown to cause an increase in the level of human beta, and it's not defensing, it's human defensin. There's no G at the end of this. Human defensin 2 levels, and I'll talk about human defensin 2 in a few minutes. So actinic keratosis, again, it's sort of this patchy, reddish, discolored, different texture type of skin, can just look like eczema for a lot of individuals. This is the most common precancer that forms on skin. It comes about as you have like chronic exposure to ultraviolet light rays, or from indoor tanning. That used to be a big thing. I don't think indoor tanning is as popular nowadays, 2025, no, probably not. And this often leads to the formation of squamous cell carcinoma, to kind of link all these things and ideas together. Again, before we go on to that though, melanoma is something that we've also talked about. There's very limited research currently, even though this is a very aggressive form of skin cancer. This is the most lethal form of skin cancer. We're not 100% sure about the types of skin microbiota that are associated more with melanoma, but it's thought that somehow a number of these different bacterial colonies may actually influence melanoma progression. There might be different therapies that different bacteria are either helping or blocking, but we don't know a lot about this. This is a huge area of research for those of you who are interested in going into it. As I mentioned to you earlier, this is the #1 type of cancer globally. This is just going to increase over time. it's a great area of research. There are more researchers kind of getting involved in it, not necessarily here in Toronto, but there are a lot of researchers in Canada, both in BC and in Montreal McGill, that are doing research into different forms of skin cancer. So maybe think a little bit along those lines. This is not actinic keratosis. In fact, this is not really skin cancer either. This is a different form of diagnostic in your skin that is also like very common. So this is called cutaneous T-cell lymphoma, or CTCL. It's not a skin cancer. This doesn't originate from any kind of progression or abnormal division of skin cells. This is a form of non-Hodgkin lymphoma, but it affects the skin. You see the results in the skin. It's a blood-borne T-cell type of thing, but actually it affects the skin the most. The type that you're seeing here is one of the different stages that you see, and this is where you have this kind of rash. This is the mycosis fungoides rash. It's a rash that often, again, looks a lot like eczema, and it just looks like a red rash. It might look like you have a food sort of allergic reaction. It might look like you have something where you've got exposure to something else, but this is persisting. It's not going to go away after a couple of days. It also has like a very rough texture, and I think you can almost see that on this figure or this picture as well. So this scaly type of rash appears on your skin, and this is not on the parts of your skin that are usually exposed to the sun. So unless you're going around trying to get a tan, you might see this on the lower part of your abdomen, you might see it on your thighs, you might see it on your butt or buttocks, as well as on the chest or on the breast area. So not normally parts that would be heavily exposed to the skin. And then the skin actually starts becoming much thinner around the rash. It might start becoming very dry, flaky, and it might start becoming itchy, much like eczema. And that's where it becomes a real issue, because how do you know? I'm not a dermatologist. Most of you aren't going to know a lot about dermatology either. And even your family doctors, they're not really trained to be able to tell the difference between like a rash versus something much more serious. So you'd have to go see a dermatologist for all of this. If this continues to progress and it's not sort of cleared up, or you're not being treated for this, this will continue to both the plaque phase and then of course the tumor phase, which is the most serious. And this is where in the plaque phase, you're going to form these small little hard nodules on your skin in those affected areas. Strangely, along with CTCL, as you'd expect, so one, your skin is literally being damaged by this non-Hodgkin's type of lymphoma, but you're also going to be changing the skin microbiome, and you are also, as a result of changing your skin microbiome, and because you are now breaking the skin, you're forming a rash, it's scaly, it's itchy, it's also becoming thinner, you're now also seeing nodules under your skin, you're also at increased susceptibility of developing infections through the bacteria that are now occurring on your skin. So these types of changes in the skin microbiome are very serious. These are ones that you as a group of students should be thinking very much about. This is also like a great opportunity. I keep saying that because I know some of you might be interested in either going into clinical practice or thinking about being involved in like research or thinking about your own future research perspectives. This would be a great way to kind of think about looking at the microbiome on your skin, thinking about how you could tackle a cancer that is so prevalent and that we don't know a lot about as well. In addition to things that we already know, the UV can directly influence immunosuppression of the skin, in your skin. The microbiome, the bacteria and fungi that grow on your skin may also help to make it even worse or suppress your immune system even more, which would then also increase skin cancer risk. So the fact that we're finding Staph aureus on actinic keratosis, as well as potentially squamous cell carcinomas, probably suggests that your skin microbiome is playing some kind of role in mediating some kind of immune effect. So this, again, lots of therapeutic implications as well. Perhaps one of the things that people could be thinking about as a topical sort of probiotic, something that you apply directly on your skin to make it helpful for good bacteria to grow on your skin, the probiotics that support the growth of different bacteria, or the prebiotics, some of the different things that it needs, the nutrients that it needs to help grow on your skin. The use of antibiotics, again, very serious. More people are taking antibiotics for all sorts of things now. It also has a direct impact on your skin microbiome as well. It's not just your gut. Your skin microbiome is also affected by antibiotic use, and possibly now the use of postbiotics, things that can be used to help to stabilize different aspects of the microbiome. Ultimately, one of the things that is like fascinating, which is part of what the Stanford group is looking at, is personalizing it. We all know that you have your own unique microbiome. It's getting easier and easier because it's the easiest microbiome to access. Like, it's easy to take swabs of different parts of your skin, and it could be possible to tailor treatments to individual microbiota profiles very, very shortly. Like the way that we can do sequencing now is so much better and faster than it was a few years ago, that it's entirely possible for you to know like the general populations that are growing on your skin. So kind of related to that, one of the things that I wanted to kind of mention, this Staph aureus is actually a really big issue, and your skin is already kind of in a place where it's trying to defend it as much as it possibly can. And why we talk about UV radiation and why we talk about things that suppress the immune system, part of that comes up here in this slide. It's great to get a tan. It's great to get some UV. It's great to just be out in the sun, especially after the long Canadian winter. But if we have the normal commensal skin microbes, the skin bacteria, and we have the normal interaction with these different keratinocytes, these keratinocytes are actually really good at trying to control the level of bacteria that grow on your skin. Your skin does a pretty good job. The keratinocytes, major cell types, we know that there's an acid mantle that already kind of inhibits too much bacterial growth, having a slightly acidic skin and production of things on the skin. These keratinocytes will also produce a protein called a defensin, or that HBD2 that I talked about earlier. These defensins, or HBD2, these defensins can act on neutrophils, and we talked about neutrophils in the deeper parts of your skin in lecture number 1. Your skin has like more immune cells than like any other single organ, so it is a very, very much an immune organ. And there is a G protein coupled receptor sitting on the surface of these neutrophils that are activated by these defensins produced by these keratinocytes. And again, the short form for this is an MRGPRA2, and that's the G protein coupled receptor. This is the long form name of this. And as a result, this activates this neutrophil to increase the production of interleukin-1 beta under normal situations. If we have too much of this Staph aureus growing in our skin, that it's overtaking the normal skin bacteria, this can actually inhibit the growth, the interleukin-1 beta, inhibit the growth of this Staph aureus and prevent it from spreading. So our skin builds up these natural sort of defenses. This defensin protein produced by the keratinocytes, again, really important. If you were to make a guess, and you're looking at the 4 to 5 different layers of the skin, what layer of the skin do you think you would have defensin being produced by? Like, which type of keratinocytes, what layer? If you're to guess. Yep. So, yep, that would be true if we're just looking sort of at the entire skin. We might see that there. But if we're looking just at the epidermal layer, what keratinocyte layer would be most likely be looking at? Sorry, I'm not sure if that means that you really want to answer or if you're kind of... Yeah, sure. Granulosum, okay. We could see them in the granulosum. That would be a good bet. Anything else? Yeah.
Maybe the stratum basale?
So the basale and the granulosum would be the 2 layers that you would most often see the keratinocytes producing HBD2 or the defensins that we're talking about here. And those are important because you understand the basic properties of the keratinocytes in those layers. I'm just going to talk about this very quickly. You don't need to worry about all the details on this slide. I just mentioned to you in the previous slide that we're so much better now in 2025 at being able to sequence the skin microbiome and even the gut microbiome. We practically have like microbiomes like everywhere at this point. But if we were to take a look, oftentimes in most of the papers that you read, you often see 16S ribosomal RNA sequencing. I think most of you have seen that in some of your other lectures around bacteria and genetic identification. One of the reasons why they use this route and they use 16S ribosomal RNA sequencing is that this 16S ribosomal RNA, the amplicons, the primer regions that we generate to amplify a genetic sequence, it actually is specific to bacteria. We don't see these regions like on fungi, and we see like 18S, 20S, and we have like different ways to amplify the signal and the genome from fungi versus bacteria. So bacteria have the 16S ribosomal RNA sequence, whereas the fungi don't, and we use different sets of primers or amplicons for these. And we're very quickly able to, using this method, just go ahead and find out very quickly all of the different types of bacteria as well as fungi and other eukaryotes that live directly on your skin. Another way is just to get a skin sample and then do whole genome metagenomics. And again, it just takes all of the different sequences at once. It doesn't care like where it's actually coming from. And then using sequence analysis, it can actually sort out whether it's a bacterial source or from other strains as well. And it's very good at doing that. This method takes a little bit longer on the right, but I wanted to give you some context. Like this is where we're sitting at in 2025. This process is actually so much faster. It's taking about 1/10 of the time that it would have just a couple of years ago. And our ability to sequence the microbiome is incredible at this point. Again, not a lot to take away from here. However, based on today's lecture, we learned about sebaceous glands, we learned about apocrine, merocrine glands, we learned about all sorts of different glands that are on different parts of the body, and that actually influences how different types of bacteria grow on different parts of the body. It's one of the first papers that we looked at, the Blazer paper back in 2005, lecture number 2. And if you remember, they sampled from different areas, from 3 men, 3 women, and they saw that there were differences in bacteria. And it kind of makes sense if you think a little bit about how they got those different results, because each of the different areas that they sampled from on the skin have very different sort of properties, and as a result of the different types of glands and what they're seeing there, the different bacteria are also going to be influenced as well. And that's really all I wanted to just highlight and demonstrate for you on here, that with these new technologies now, we can actually go back and verify some of those original papers from back in 2007. They all seem to be very correct. There's really no difference, and we're able to do it much faster, and we understand the biology behind why we get these differences in the different kinds of bacteria in the different regions of the body, and hopefully the part of the lecture on the different glands kind of makes sense. The last thing I'll sort of talk about on this slide, just for I guess my verification that you sort of were paying a little bit of attention, and you can always look back in your notes as well. These are the most common types of bacteria that they found here on this particular updated study from 2023. Are these the same overall types of bacteria that were found in the original Blazer paper, like Propionibacterium? Did they find that originally? Corynebacterium? And did they also find Staphylococcus? Do any of these ring a bell? I will also bring a bell for the next lecture. So all of these should, because they were found in the original paper, again, verified in this paper here. There's one other, I don't know if you remember it. Starts with like an F. Yep. Okay, Feingolia, yeah. So that was another one, A something something, I forget the whole name, but generally speaking, again, one of the things that's really remarkable, when you think about the technologies we have now versus what they had back then, the sequencing data actually turns out to be very, very accurate, and it's actually really interesting. The fact that we also have like human papilloma virus growing in different parts of our body, when we talk about cervical cancer toward the end of the course, just keep in mind that human papillovirus is also something papillomavirus is also something that we can detect. When we talk about cervical cancer, this will be a big issue when we go back and talk about it. Again, I'm not going to make too much of a deal on this, just to kind of indicate for you that the bacteria on our skin do a number of really different things. Not just the skin cells themselves, bacteria also want to prevent other bacteria from over-colonizing. So we really don't want too much of that Staph aureus growing around. We know that the dominant sort of skin bacteria is the propionic bacteria that we just looked at. It will produce these small molecules like CP3 that you see here, and I'll often abbreviate it as CP3. One of the interesting things that this molecule does, it actually enhances the ability of the Staphylococcus to actually form a biofilm. And I don't know if you remember your biofilms from like second year health and disease courses. I think you guys have all learned that. Biofilms are good or bad in general, or do you care? They're okay. They're like not terrible, they're not bad, they exist and they grow. Part of the reasons why we care about them is because they have very different properties than individual bacteria. They prevent other antibiotics from getting easy access, but they also become substrates for other bacteria to grow on them. And one thought is that the propionic likes to actually have these biofilms because they can colonize on top of them. So they're kind of using it to their advantage by producing this CP3. And again, that's the only thing I want you to take away from this. We've already talked about the fact that actinic keratosis is sort of a pre-cancer, and these pre-cancers are often associated with the Staph aureus. It's not a good bacteria to have like growing in large numbers, and we see this on your skin, and increasingly it's thought that if you have too much of this Staph aureus around on your skin, that it will eventually lead to things related to actinic keratosis, which is the precancer, or the actual basal cell carcinoma, or the squamous cell carcinoma. It actually does increase the odds ratio, making it more likely that if you have those cancers, that you have like Staph aureus associated with it. Right now, it's very correlational. There are very few studies that have actually directly shown that if you have an overabundance of this, that it can actually produce cancers. But in animal models, it does seem to be the case where Staph aureus can actually induce skin cancers. And we'll talk about that in a future lecture. This just, again, is the defensins that are being produced, and they seem to be one of the big sort of things that people are looking at right now in terms of research. I think I'm just going to give you a very quick overview of this, and then we'll kind of wrap things up next week. The second half of next week's lecture, I'll leave it up to you. So review, a little bit of a review. What else do you want in the second half of next week's lecture? Because the first half we'll just go through this paper and talk about it, model for skin cancer. What else do you want to have? Do you want nothing? No? Okay, so we'll go through some practice questions, sure. Okay. Anything else? No. Yeah, please.
One of the questions in advance, if we do good on the practice question, maybe two.
Do you guys want some questions that you'll actually get on the term test in advance? That's what your colleagues were asking.
Maybe one, yeah.
Maybe one? Sure.
We're not greedy at all.
Okay, not greedy. Okay, yes, please.
Maybe even like methods on how to like answer the short answers, or like even like keywords that you look for.
Okay. So a marking scheme related to the short answers?
Yeah.
Okay, sure. Yeah, please.
Maybe could we also have example answers for the short answers? What a answer would look like?
No, I like that. I like that. But you know what? I would always mark myself like really easily. So I'm always going to give myself like full marks. So could I make a counter proposal? If I give you a couple of like sample short answer questions, would some of you be willing to kind of write out like a short answer and then send it back to me, and then I'll anonymize it, take it up and show you like how I would have marked it? Would you be okay with doing that? Okay, so that's easy. Yep, done. We'll do that. Anything else that you guys would like for next week? Are you at all concerned about your term test? Not really. No, good. Feeling good? Not feeling good? I don't know. Can't tell. Okay, so this is what I propose that we do for next week, since you guys suggested it. So the first hour, I am going to take you through some of the major parts for this paper. And just again, to give you a little bit of a breakdown of how you should be studying as you start studying. Sorry, I still have like 2 more slides to go, and then we'll kind of wrap things up. So one of the things that I told you earlier, you're going to get more questions from this week's lecture. So if you're going to go and study this week's lecture, last week's lecture, and then like the other 2 or the other 1 lecture. Next week's lecture is going to be a little bit dense, but you're still going to get about 3 multiple choice test questions just from next week's lecture. If you have questions in class, break the mold. Ask a question in class, because you're not going to have that time to be able to ask like in the week that's coming up. It won't be as easy for you, so like ask questions in class. And in the second hour, what I would propose that we do, I will give you like a very, very quick review of like the major things that you should be focusing on from each of the different lectures. I will also provide beforehand some sample questions. You will provide me with some sample answers that I will use in sort of that review as well. And then I'll give you sort of a condensed version of all the slides. Like, I think we will have roughly about 130 slides. Do you need to know all of them? No, you probably need about 65 to 70 slides, core slides, to do like an A or A+ on the term test. I'll give you that in advance of next week's class, and we'll use that sort of as part of the review. And since you were so nice to ask, I will give you, how many multiple choice is it? It's 15, right? I'll give you 3 of your multiple choice test questions in advance, and I'll give you 1 of your short answer, parts of your short answer in advance as well. But you guys will have to do a lot of talking. Fair enough?
Yeah.
Okay, great. So next week, I'm going to be talking to you about this paper. So we've talked about the fact that you have bacteria that grow on your skin. We talked about the fact that you can disrupt your skin. And it turns out that if you keep damaging your skin, it's not good for you. So actinic keratosis, not good. Dry, scaly skin, not good. And one of the reasons why dry, scaly skin is not good is because once you damage your skin barrier, bacteria love getting under your skin and start growing there. And it turns out that all the things that we've been talking about, the immune system, the different levels of layers of the skin, the bacteria that grow on your skin, and all of those different factors are going to be coming together. So when we start having damage to the skin, one of the things that can happen is it can heal completely, and that will actually be part of the very first lecture. And one of your one of your multiple choice test questions will come from this, where it's going to be just how does the skin normally repair itself, and you'll be able to answer that question easily. However, if you keep damaging the same area of your skin over and over again, and it's healing but not completely healing, eventually on your skin you're going to start forming an ulcer. And that's unfortunately what happens in the next module in diabetic neuropathy in your skin. You actually form an ulceration. Your skin actually really suffers from diabetes and having too much blood sugar. And one of the things that happens often in diabetes, first thing that you see is like a skin ulceration. The skin just doesn't look good, it starts breaking down, and you start getting this ulcer. If this doesn't heal, and you keep getting this ulcer and you keep damaging that part of your skin that has the ulcer, which again, for many people with diabetes, often their feet and legs, basically that part of your skin becomes cancerous. And one of the things that happens is this ulceration can become a form of squamous cell carcinoma, which is called Marjolin's ulcer. And this is where bacteria are now going to be involved, and this is where also the immune system is going to get involved. And this, again, was one of the first papers that came out in 2014 to link the immune system, skin, and skin cancer formation. And Marjolin's ulcer is a variation of squamous cell carcinoma that we'll be talking about in the next lecture. So hopefully all of that will come together for you, and I'll look forward to lots and lots of interaction in the next class, so much so that I won't have to bring a pin or anything else. Otherwise, I will have office hours on Friday at 1 o'clock over in college. You're welcome to drop by at any time, and if I don't see you before then, have a great rest of the week, and I'll post this lecture recording as soon as it becomes available.